The overall goal of this procedure is to measure the levels of 24 plasma circulating microRNAs in order to determine the risk of developing lung cancer in heavy smokers older than 50 years.
This method can increase cost-effectiveness of low-dosed computed tomography screening programs for early lung cancer detection by reducing the number of false positive nodules and possibly overdiagnosis.
The main advantage of this technique is that circulating microRNA are easy to measure with standard equipment adopted in common molecular laboratories.
We first had the idea for this method when we began to study the role of tumor microenvironment on the development of lung cancer. MicroRNAs reflect the host response and the dynamic interaction between the tumor and its microenvironment.
Visual demonstration of this method is critical to better understand standard operative procedure to follow for this type of molecular analysis.
To start this protocol, use vacutainer tubes to collect 10 milliliters of whole blood sample. Store at room temperature.
Do not store whole blood at low temperature, such as 4 degrees Celsius, to avoid thermal shock and cell lysis that lead to an unspecific microRNA release.
Centrifuge the plasma within 1 hour to separate it. While making sure to avoid contact with the lymphocytic ring, transfer the supernatant into a 15 milliliter tube. Then, centrifuge the supernatant at the same conditions. Aliquot 1 milliliter of this plasma supernatant into 1.5 milliliter cryovials, being careful to avoid collecting the plasma fraction from the tube.